आज हम लोग पढ़ रहे हैं सिंथेसिस, केमिस्ट्री ऑफ डीएनए सिंथेसिस। ठीक है, मॉलिक्यूलर बायोलॉजी में है आप लोगों का, तो इम्पोर्टेंट तो है केमिस्ट्री ऑफ डीएनए सिंथेसिस। ठीक है, तो ये जो केमिस्ट्री ऑफ डीएनए सिंथेसिस है ये इम्पोर्टेंट है। तो इसमें क्या लगता है केमिस्ट्री ऑफ डीएनए सिंथेसिस में? व्हाट आर द मोस्ट इम्पोर्टेंट एंजाइम्स दैट आर नीडेड इन नॉट एंजाइम्स, व्हाट आर द मोस्ट इम्पोर्टेंट थिंग्स दैट आर नीडेड इन टू थिंग्स, DNTPs, this DNTPs, all four types of DNTPs in excess are needed to complete the DNA synthesis. So DNA TPS, DTPS, DCTPS and DGTPS, चारों के चारों बहुत ही इम्पोर्टेंट है फॉर द केमिस्ट्री ऑफ सिंथेसिस ऑफ डीएनए। ठीक है, नेक्स्ट हम लोगों का जो इम्पोर्टेंट है, नंबर 2, दैट इज प्राइमर, टेम्पलेट, junction. Primer, template, junction. What is this? Shorter RNA primer is extended into long DNA. Okay, shorter RNA primer is extended into long DNA. तो एक शॉर्टर आरएनए प्राइमर लगेगा, प्राइमर टेम्पलेट जंक्शन में लगेगा, और एक्सपोज्ड 3' OH एंड 3' एक्सपोज्ड रहना चाहिए, 3' OH एंड फ्रीली ओपन रहेगा, एक्सपोज रहेगा, नॉट म्यूटेटेड, नॉट एनीथिंग डन, एकदम 3' OH शुद्ध फॉर्म में एक्सपोज रहेगा। ओके, अब हम लोग देखेंगे डायग्राम। आप लोग चाहिए तो 2x में देखिए वीडियो, अगर चाहते हैं तो थोड़ा सा इसे खींच के बड़ा कर लीजिए, वीडियो को एक्सटेंड कर लीजिए, ये पार्ट स्किप कर दीजिए, पर आई विल ड्रॉ इट वेरी नाइसली। जो स्टूडेंट्स चाहता है कि ओके कि कैसे ड्रॉ करता है देखना, और जो स्टूडेंट चाहते हैं कि लास्ट में एंड में देखना कैसा ड्राइंग है, तो दे कैन डू अकॉर्डिंगली, बट आई विल ड्रॉ इट ओवर हियर स्लोली वेरी नाइसली। ओके, ये मैं अभी टेम्पलेट स्ट्रैंड को ड्रॉ कर रही हूँ। ये है मेरा टेम्पलेट स्ट्रैंड। ये क्या है मेरा टेम्पलेट स्ट्रैंड, इधर मेरा 3' OH strand exposed. Now this is my template, you can mark it over here smallly, ये template है। Okay, now I am drawing the primer. प्राइमर क्या है कंडीशन? शॉर्ट होना चाहिए। ये मेरा शॉर्ट प्राइमर है, जिसका OH प्राइम एंड इधर है, 5' एंड P के साथ इधर है। ओके, ये लाइन थोड़ा सा ऐसा होगा, ठीक है। तो यहाँ पे मेरा A है, यहाँ पे T है, यहाँ पे G है, यहाँ पे C है, यहाँ पे A है, यहाँ पे E है, यहाँ पे C है, यहाँ पे G है, एंड दूसरा अभी यहाँ पे आएगा इस लोकेशन पे। यहाँ पे तो हाइड्रोजन बॉन्ड फॉर्म हो गया, यहाँ पे भी हाइड्रोजन बॉन्ड, यहाँ पे भी हाइड्रोजन बॉन्ड, यहाँ पे हाइड्रोजन बॉन्ड फॉर्म हो गया। अब नया डीएनटीपी आ रहा है, नया डीएनटीपी आ रहा है जिसका ऑब्वियसली OH है इधर और 3 फॉस्फेट है, ठीक है। अल्फा फॉस्फेट है, बीटा फॉस्फेट है और गामा फॉस्फेट है। 3 प्राइम OH जो है, this OH will react with the alpha and यहाँ पे फॉस्फोडाइस्टर बॉन्ड क्रिएट होगा by SN2 रिएक्शन। यहाँ पे क्या फॉर्म हो रहा है, फॉस्फोडाइस्टर बॉन्ड फॉर्म होगा and this two will be released, that is phosphate beta and phosphate gamma. These two are called pyrophosphate and over here ये रिलीज हो जाएगा। मतलब रिमेम्बर रखना मेरा कितना स्ट्रैंड था, 1 2 3 4 5, तो 1 2 3 4 तक था, 5 में आ जाएगा, ठीक है, नया डीएनटी। तो यहाँ पे सपोज C था तो G आएगा, उसको देखते हैं, ठीक है, उस डायग्राम को देखते हैं। तो कैसा होगा, SN2 रिएक्शन हुआ, वहाँ तक समझ में आया, ओके। ये है 5 प्राइम वाला एंड, 5 प्राइम, और दिस इज 3 प्राइम OH. Now RNA primer is coming. It will start from here, but before the RNA primer, one thing, write all this A, T, G, C, A, suppose. So till 3 last time we have done, so the 4th one will be introduced, but you can write over here G, C, this way it is going on. So when the new primer comes, this way, so this is the primer and this end is introduced, again the 3 prime OH end will be this side, 3 prime OH, and this is the 5 prime. So you can understand the difference. This till here we had done last time, but the new DNTP came and introduced this, so T will come from this side. So T will come from this side, and this portion we have now done. Same way another will come, okay, another PP will come, triple P. Over here there will be OH, so that will again be introduced in the same way. I hope this is clear now, by now. And what will be released? Pyrophosphates, that is phosphate beta plus phosphate gamma. These are pyrophosphates. What happened here? What reaction happened here? This is SN2 reaction occurring over here, and O is reacting with the alpha. This OH is reacting with the alpha P and forming the hydrogen bond and bringing the primer. This way it will keep on extending. So till here is double stranded DNA, till here is double stranded DNA, and the other part is single stranded DNA. Clear till here? This way it will go on the process, and this way it will happen, chemistry of DNA synthesis. I hope it is clear. Okay, this one is the template strand, this one is the RNA primer. Okay, fine. Now let's write the reaction then. you have to explain this in this way only in exam, but for more reference I am doing this. Okay, so at first you have DNMP, n number, then new DNTP is coming, then you are getting DNMP, which is n plus 1. Am I logically correct? Am I logically correct? Obviously 1 is increasing at each step. We increased 1, there was 3, then 4 double stranded became, and other is the single stranded DNA still now. So 1 1 will increase, and we are getting pyrophosphate, phosphate, beta phosphate gamma. This is pyrophosphate. What is this? Pyrophosphate. This is your chemistry of DNA synthesis. I hope I am clear. DNMP was there, monophosphate, then triphosphates came, then monophosphate plus 1 it got it, and then this is the overall reaction of chemistry of DNA synthesis. This is the overall reaction of chemistry of DNA synthesis. Am I clear? Am I clear? Okay. So this way you can show one step or two steps. Now we have few questions related to this. We will solve that. If you want, in today's class only we are going to do, and if you are tired students, then please let me know, message me in WhatsApp, then I will not do. Okay, so tell me. Okay, fine, let's do then. Okay, then we have overall reaction. Okay, over here, this reaction, the delta G of the reaction, the overall reaction you have written, everyone. Let me erase it. Okay, so the delta G is slightly negative, 2.5 kilocalorie per mole, slightly negative. And if it is not much more negative, then DNA synthesis will not be smooth. Okay, so what happens if not increase this slight negativity, then the spontaneous reaction will be stopped. Smooth synthesis of DNA will be stopped. How to overcome? We do pyrophosphatase. Then this we do pyrophosphatase. Okay, what is this pyrophosphatase? This pyrophosphatase question can come. What is the role of pyrophosphatase? I will explain you very nicely. Concentrate here. What is the role of, not pyrophosphate? It is pyrophosphatase. Pyrophosphate we are getting, but what is pyrophosphatase? Okay, what is the role of pyrophosphate? Note down this question. Okay, so students who are watching this video online, मैं माफ़ी मांग लेती हूँ क्योंकि मेरा ऑनलाइन क्लास चलता है, तो उसमें मैं रिकॉर्डिंग करके देती हूँ. It's like I have more students sitting over here. All of them are watching this and doing with me. Okay, and obviously over here I have one student which I am writing on my phone. They are writing it on the whiteboard. Other students are copying it. Okay, I only write on the screen for you students, YouTube students. So please mercy me for the lengthening of the video, excess talking between the video. You can skip those parts. You can just speed up the video to 2x and then watch it. But I hope you like my explanation. Therefore, you are watching my video. So yeah. So what is the role of pyrophosphatase? So the delta G of the reaction, what was the reaction? Do you remember? I am writing it again. D N M P. Suppose we had N plus, obviously plus mu DNTP is coming. Then we are getting DNMP in each step. One is increasing, so N plus 1. And along with that, we are getting P. Okay, they are linked, and these are called pyrophosphate. Okay, note it down till here, then I'm explaining. Give me 1 second. 1 second. You note it down till here. So, till here, understood? Okay, this pyrophosphate. And the overall delta G, you know what is delta G? Activation energy? No. Gives free energy. The whole delta G of the reaction is minus 3.5 kilocalories per mole. Kilocalories per mole. Isn't it too less? Isn't it too less? Isn't it too less? Yes? Yes. It is too less. So, let me. Okay. So, if it is too less, if the delta G remains very slightly, if question comes in exam, this way you will write slightly negative, then smooth synthesis of DNA will be hindered. Smooth synthesis of DNA will be hindered. If minus means negative is not increased, then spontaneous reaction will be stopped. Therefore pyrophosphatase is required. Pyrophosphatase is required, which converts pyrophosphates into 2-pi required. That does what it does. Pyrophosphate to 2-phosphate group and the delta G every time is increased by minus 7 kilocalories per mole. This allows smooth running or smooth synthesis of the DNA. Am I clear? Am I clear? Okay. Today, keep this video till here only. Next class, we will go in the role of two metal ions. DNA polymer is distinguished, and from bone model. Obviously, very important.